Good evening, all the participants and resource person Rahul Kumar Ji. I welcome all the participants in the Science Dialogue series. Most of you are knowing the objective of this Science Dialogue series. Those who are new, for them, this is free Science Dialogue series, webinar series, in which we invite resource person from different institute who has good reputation in different domains of the biology. So neither we charge any fee nor we offer any remuneration to the resource person, but resource persons are very known. Those who have good reputation, they only deliver lecture, and more than 100 lectures has been organized under Science Dialogue series. After few lectures, we have started giving e-certificate also if people attend complete lecture and fill the feedback form. But we share that feedback form during lecture, means yes, during lecture, after the last part of the lecture, in the chat box here only, not in the different groups. And so purpose is to disseminate quality knowledge. We have 140 crore population, but you know that we are begging something scientific thing from European country, American country, and different countries. So we are trying to bridge this gap. In most of the times in private university, state university, there is a scarcity of teacher, and most importantly, the scarcity of good teachers. So through this you can say way, we try to improve the overall knowledge. We are trying to improve the knowledge pool of the society. 2 days in this series, our resource person is Dr. Rahul Kumar, and he is assistant professor at Magadh University, one college, that constituent college of the Magadh University. He is very dynamic. We both are from same PhD from same institute, that is Jawaharlal Nehru University. He did his PhD from School of Life Science, and I have seen him very active in different platforms. And he's also trying to disseminate his knowledge to improve the knowledge of the society. That's why he's here. And participants, Rahul ji, participants are not only a student, but faculty also, researcher also. Those who wish to attend and gain the knowledge in that topic, they all are welcome to attend. So with this brief introduction, we welcome you in the Science Dialogue series. And we are also grateful that you are ready to share your knowledge. You are ready to give your time, because everybody is busy in his or her scheduled or paid things wherever they are. They are very busy in their work or some family work also. So please, now platform is yours. Unmute your mic. Yes, yes, Rahulji.
I'm going to share my screen.
Yes, yes, yes. Ashish ji, Abhishek, please close your camera.
Sir, is my screen visible?
Yes, yes, the screen is visible, very well visible.
Okay. Thank you, sir, for invitation.
आपके लिए कि वॉइस थोड़ा बढ़ पाए तो थोड़ा सा देखिए ना.
Yes, sir. Sir, it is audible now?
Now it's audible, yes.
Okay. First of all, I would like to thank respected sir for inviting me for this great talk series, that is Science Dialogue series. And when I was at School of Life Sciences, I was working on molecular biology. Then I have worked at various institutions. I have worked at AIMS New Delhi. I have worked on science education also at NCRT. Then I worked at ICR, IRI, New Delhi. There I started working on taxonomy. And then currently I am also working on taxonomy while working as assistant professor at Bihar. So the topic on which I am going to talk today is species concept conundrum. and integrative taxonomy. So, what is conundrum? What do we mean by conundrum? Conundrum is something which is confusing, which is unsolved. So, in this lecture, I will focus on species concept, what challenges it posed to the taxonomist, and how it is contentious still today, because we consider taxonomy as a classical science, but this classical science is still relevant and still posing so many challenges. We will look on all these issues one by one and how these challenges can be solved by integrating multiple disciplines in studying taxonomy. So, this brief intro, I would like to start with a story. This story dates back to 2019. That time I was working as a DST Surf Fellow at Division of Entomology IRI, that is Indian Agriculture Research Institute. There I was working on tortoiseshell moths. Tortoiseshell is a family of moths. Moths, you must be knowing difference between moths and butterflies. They belong to the same order, Lepidoptera. So these moths are very small in size. They are bell-shaped moths. So I went to Meghalaya and Nagaland to collect those moths. But one of my friends, he was my colleague at IRI, so and he was not able to visit along with us for the sampling. So he requested me to bring some ants because he was working on ants. So what I did, I was doing my own work also because he requested me. I was collecting ants for him only, and interestingly, I found a star fruit tree, and there was some yellow colored, bright colored fruit was hanging from the tree. So I plucked that fruit and I brought it to my guest house. I was staying at guest house there, and when I carefully looked at that fruit, I was going to wash that fruit. So I found some cottony structure present there. When I scratched that structure, I found a small ant coming out of that cotton like structure. So I thought that means I don't know why, but I did not kill that ant, and I collected that ant live. And the rest of the ants I collected in 70% ethanol. People who study insects, they must be knowing that small insects, they are collected in 70% ethanol. Like we use formaldehyde for large sized animals, we use 70% ethanol for small sized insects to preserve them.
Sir, hello sir, can
So
हाँ
I
बोलिए प्रिंस क्या है?
सर क्या आप मतलब हिंदी इंग्लिश में हल्का बोल सकते हैं सर? हिंदी इंग्लिश में प्योर इंग्लिश बोल रहे तो पता नहीं चल रहा है सर।
नहीं, यहाँ तो इंग्लिश के लिए है। आपको कुछ ऐसा चल रहा है तो आप हिंदी में हिंदी का अलग से करेंगे प्रिंस जी।
ओके सर,
क्वेश्चन आंसर में आप हिंदी पूछ सकते हैं, है ना?
ओके सर, आई अंडरस्टैंड। ओके सर,
okay, people are facing some difficulty, so I can switch to Hindi also in between. So
हाँ, इन बिटवीन ठीक है, स्पीकर के उस पर में, बट नॉट बाय चॉइस, है ना? ऐसा नहीं।
यस
इंडिविजुअल
सर,
देन
इन बिटवीन।
डिफरेंट पीपल विल अगेन आस्क in different language. Some people will ask in Mathli, some people in Marathi. So
नो नो नो
language,
सर, इन
हाँ
बिटवीन
हाँ,
वी।
क्वेश्चन आप पूछना प्रिंस कुमार। यू कैन आस्क क्वेश्चन एंड समटाइम्स ही फील कि कुछ चीज जो है हिंदी में करना है, सो इन बिटवीन ही कैन स्पीक इन हिंदी आल्सो, है ना?
ओके सर,
जी जी।
then what happened? I collected that ant alive and I brought it back. After returning back, I was observing it under the microscope and I was just taking photographs. So one of my friends, her name is Dr. Stuti. She did PhD from BHU. So she saw that those photographs and she told me that these are myrmecomorphs. So I was confused because this term was new to me. So I asked her what is myrmecomorph. So she told that Rahul, this is not ant. This is a spider. It mimics ants. So
Dolly Sharma, mute your mic,
so such
otherwise I will remove you.
such arthropods which mimic ants, they are called myrmecomorphs. So I was fascinated with that thing that there are so many species, they are mimic, they look like other species, and not only that, they are non-mimic also, they look alike. So this interesting this interesting story, it happened in actual when I was at Nagaland. It fascinated me to work more on taxonomy and biosystematics. So before going further, I would like to give a is high school knowledge about some basic terms like biosystematics. So it is scientific study of the kind and diversity of organisms and of any and all relationships among them. So the relationship term is very important there. This relationship is mostly evolutionary. And the field of systematics has two major components, that is taxonomy and phylogenetics. We know taxonomy is the science of naming of living and ancient organisms, and phylogenetics is the study of evolutionary relationships among organisms. And the modern systematics is called biosystematics, and its synonyms are like new systematics, even experimental taxonomy term we use. And many scientists they consider the goal of biosystematics is to investigate the origin of organic diversity. That is, it is related to evolution. And biosystematics consider species as dynamic units. Means species are not a static unit because species keep changing. And the modern taxonomy is integrative in nature. Like classical taxonomy, we call taxonomy as a historical science, ancient science. No, it is not like that. Modern taxonomy is there and it is integrative in nature. And modern classification is based on multiple evidences like paleontology, embryology, anatomy, as well as molecular biology. So integrative taxonomy is based on multiple and complementary perspectives. So when we talk about biosystematics, we talk about modern taxonomy. The phylogeny is intrinsic part of it, and phylogeny is represented using a phylogenetic tree. So what is a phylogeny? It is a chronological account of the events by which species or other taxa have sequentially emerged from a common ancestor. For example, I have shared a photograph of a typical phylogenetic tree, and its part, the base is called root, and there are branches, nodes. So all these depict the diversification of different taxa, and it is related to the root. So root is the common ancestral taxa which gives rise to the higher forms. And modern taxonomists initially they used to prepare a character map, means they used to tabulate character. They used to give weightage to each character, and through that weightage they used to construct a phylogenetic tree manually. But because nowadays we have molecular data with us, so modern taxonomists are using DNA as well as protein sequences to construct phylogenetic trees. So this approach is called molecular phylogeny. So, if you wonder about phylogenetic tree, its history, it is very interesting to quote to mention Darwin. The first phylogenetic tree was actually drawn by Darwin. So, it is the photograph of the original phylogenetic tree. So, it was not a real phylogenetic tree. He was trying to show how animals evolve because the branch length is same and this is arbitrary. But he was the one which gave concept of phylogenetic tree. But the real phylogenetic tree, the real phylogenetic classification, it was put forth by Willy Hennig. Willy Hennig was a great entomologist. So if you wonder, it is very interesting to quote that he was the one who put forth the concept of phylogenetic tree in scientific manner, that too among arthropods. So this also makes arthropods an interesting group to study. Therefore, I would like to give a brief overview of phylum Arthropoda. It is the most diversified phylum in the animal kingdom, and more than 1 million species are known in this phylum. And it evolved about 540 million years ago. So it is very ancient group. It is very, very, very ancient even than us. And these are primitive yet evolutionary most successful taxa. We know cockroaches are in every household. There are mosquitoes, and these are in this form for million of years, and they have not changed so much because they are endowed with innumerable adaptive features and they are cosmopolitan in occurrence. And modern classification classifies arthropoda in four, because the purpose of modern classification is not to classify correctly but also to simplify the classification and not complicate it much. So class Arthropoda is now divided into four subphylums: Hexapoda, Myriapoda, Crustacean, Chelicerata. And in this presentation, I am going to focus on mostly terrestrial arthropods. They mostly belong to class Insecta, class Arachnida, and subphylum Myriapoda, particularly Chilopoda and Diplopoda. So the central theme of this talk is species detection and species delimitation. And species concept is one of the most controversial concepts in systematic zoology, and species concept problem is closely linked to the issue of species delimitation, how we will see. So first of all, I would like to define species. So there are many concepts like phylogenetic and other concepts. They define a species on uh, please mute yourself if you are unmuted. So the biological species concept was proposed by Ernst Mayr. It is the most popular and widely accepted species concept in biology. So according to biological concept, a species is a group of organisms that are capable of successfully interbreeding to produce fertile offspring. When we use the term interbreeding, it means two different member of a population can interbreed and produce offspring which is fertile. So it has a behavioral aspect. When we say interbreeding, breeding is associated with reproductive behavior, so it has a behavioral aspect. So keep this in your mind. We will discuss it later, how behavioral studies are important in species delimitation. So before that, we are going to see some of the issues with species delimitation. For the most simple way is to look at the morphology. So if they are identical, we consider them same species. Like in the first column, first row, you can say, see, I'm going to switch on the pointer option. So as you can see my pointer, the first row, all I hypothesize they all belong to the same species based on the morphological characteristics. The first column, first row, species A and species A. They are from the same species. They have identical morphology. Then there is one group where I have drawn a bigger circle and a smaller circle. They also belong to the same species, because sometimes phenotypic plasticity, which may be seasonal, morphological changes, there may be some environmental factors which lead to appearance of a specific trait, a specific characteristics in particular climate, particular season. So it leads to creation of a different phenotype. So it leads to differences among the members of the same species at different geographical locations. So this second column and second row and the third column and the third row, you can see these both indicate same species, but they are morphologically different. Similarly, um, two species may look identical, they may look different, and they may be different both genetically and phenotypically. So there are multiple probabilities, and there are multiple chances, there are multiple options there where we get confused, where we are not able to uh, correctly delineate a species. So the International Code of Zoological Nomenclature and scientists, they have put forth some basic principles to classify species. First, we look at number of dissimilarities and similarities. So if the number of dissimilarities are more than number of similarities, then we tell that yes, these two are different species. Because in taxonomy, we collect dead samples, because it is not always possible to collect a live sample. So I will come on that also, why collecting live sample is important. So we collect dead samples. So how we will tell that these are able to interbreed, because there are many species which look identical but they are different. So how we will study interbreeding? So we look at their reproductive structures. If their reproductive structures are identical, we tell they belong to the same species. Next, it is the nowadays very popular molecular taxonomy and phylogenetics. We sequence a small portion of DNA from animal or plant, and we compare to multiple sequence alignment. Then we create trees to understand if it is the same species I am looking for or it is different. And behavior. And behavior is very important in studying interbreeding, which has been ignored for by taxonomists. It is mostly ignored by taxonomists because taxonomists are mostly concerned in First of all, their main motto is to identify and discover new species, and next they mostly focus on the morphological differences, and now they are also going for molecular taxonomy. And with invention of modern easy sequencing facilities, advanced sequencing facilities, next-gen sequencing facilities, they are going for whole genome sequencing, but it is quite costly and it requires lots of funding. So it is still not feasible in a country like India. So behavior and other aspects, they are of great significance. So before moving further, because I already discussed there are issues associated with the species delimitation. So what are the issues we will see in this example of species complex? So a species complex is a group of closely related organisms that are so similar in appearance and other features that the boundaries between them are often unclear. So what do I mean here? So I have given here three examples. The first example is a Bimesia tibesi. Bimesia tibesi is called whitefly. Commonly, its common name. It is a serious pest species, agricultural pest, and it is widespread all across the world. So it is cosmopolitan in occurrence. So here you can see, this is the cluster map. You can see different clusters. There are different biotypes of Bimesia tabaci. Bimesia tabaci is the single species. They are morphological. All these clusters are morphological same. They are genetics. Their genetic makeup is same. Still, they are not able to breed among themselves. Why? Why there is such reproductive isolation? And this isolation is based on the commensal bacteria present in their gut. This is a unique case where the bacteria present in their gut is determining whether this biotype is mate with other biotype, or the population of a same biotype, their individuals will be able to mate among themselves. So these are called biotypes, and this is determined. So you can see species delimitation is how confusing and it is, how complex, and how it is still a conundrum, which is also there in the title of this talk. So apart from Bemacetaceae, here there is an example of Korean leptonated spiders. So here you can see all these spiders are of different species, but they look identical. So how they have been differentiated? They have been differentiated based on their male reproductive structures, differences among male reproductive structures. And this is the last case, last example, not last case, where you can see all these butterflies are of the same species. They can mate among themselves, and but they live in different geographical locations, and they have developed phenotypes which are different from one another. And this is very common in many papilio butterflies, and this is called polymorphic mimicry. And all these are female. You can see all these butterflies are female, and the lower right, on the extreme right, the yellowish one, this is the male. So it is called sex-limited mimicry. It is very common in papillary butterflies, and it also create problem in identifying species because they look altogether different. So one may confuse them with different species. So the existence of extremely similar species may cause local and global species diversity to be underestimated. So what is the solution? The solution is that they should be identified by rigorous studies of differences between individual species using morphological details, behavioral studies like test of reproductive isolation, DNA-based methods, molecular phylogenetics, and DNA barcoding. So in my study, which I'm going to present as a case study of this talk, so it deals with with a species which exhibits mimicry. So what is a mimicry? So mimicry is imitating other animals which are mostly harmful, and by those animals which are harmless. And mimicry is an evolutionary strategy where an organism imitate other species to gain a survival advantage. And mimicry is the product of co-evolution. Co-evolution means if different animals, different species, they are living together, they develop some adaptations to adapt among themselves. So this is called co-evolution. And because evolution takes time, and if you want to see the example of evolution, so mimicry is the best example to see evolution happening in nature. That's why Ronald A. Fisher has aptly called theory of mimicry as the greatest post-Darwinian application of natural selection. And mimicry has some purposes. The major purpose is to avoid predator first, second to attract prey, and third to enhance survival. And mimicry has many types, but the main types are Betson mimicry, Mullerian mimicry, and aggressive mimicry. In this talk, I will be dealing with Betson mimicry. What is Betson mimicry? In Betson mimicry, a harmless mimic mimics a harmful model, which is often obnoxious. You can see here, this is the American coral snake. It is very poisonous. And this is a snake. It is called, its name is Lamp Lampropeltis. It is a difficult name. So it is non-poisonous, but it mimics the color pattern of this poisonous insect. And by mimicking it, it gives signal to its own predator that don't attack me. I am also poisonous. So by adopting such strategies, the harmless animals also get benefit from their imitation. These benefits are survival benefits, which ultimately lead to their establishment in a particular habitat, which has long-term evolutionary gains. So there are some inaccurate and accurate mimics also, because some mimic, what happens, some mimic, they do not completely mimic some model, but they partially mimic. They have some other benefit. There are polymorphic mimics, like here, you can see here, you have already seen in Papiliodardanus. So this is example of polymorphic mimicry. There are sex-limited mimicry. This is also example of sex-limited mimicry. Here only females are mimicking. There are chemical mimicry also. And why I'm calling mimicry? Because these all these butterflies, they are of different phenotypes. In their particular habitat, they are mimicking some other harmful butterflies, some poisonous butterflies. That's why I'm calling it polymorphic mimicry. There are chemical mimicry also. The animal may not look like a mimic, but it may mimic the molecules secreted by some harmful insects. Like there is a moth called death head moth, because there is a structure of a skull on its thorax, and it mimics the smell of bees, honey bees. What does it do in night? It enters the honeybee hive, and they steal their honey. And during night, honeybees, they cannot see, they are only dependent upon the smell. So because they mimic the scent of honeybees, so they easily steal the honey without getting detected. So this is called chemical mimicry. There is one Westmanian mimicry. In Westmanian mimicry, what happens? The mimic itself will uh, dictate the model in such a manner and create such a signal that the model comes to it, and it will consider it like its own sibling, and it will try to feed the mimic, considering it the part of its own family. So it is called Westmanian mimicry. And there are mimicry rings. Like it is not like one mimic is isolated from other mimics. There may be different mimics mimicking the same model, and there may be possible that different mimics are also mimicking one another, as you can see in Mammalian mimicry. These are called mimicry rings. So with this fundamental introduction, I would like to introduce you to polymorphic mimicry. I have already described it, but this photograph I have taken from a paper of Kunte sir, Krishna Kunte. He is a famous scientist working on butterfly. He works at NCBS Bangalore. I got opportunity to meet him in one of the workshops. So these are all polymorphic mimic. You can see here, this is Hypolimnus misipus butterfly. So the male looks like this, but female look like this. You can see all these are all these are female, and they are mimicking the butterflies. The model are shown on the right side, and the mimic is shown on the left side. So these females in different habitat, they mimic different models. So such kind of mimicry is called polymorphic mimicry, where single species in different habitat, they mimic different models. Hope it is clear to you. So the basic questions which I'm going to deal with in this lecture. So the first question is, why does polymorphic mimicry evolve? I think till now, the discussion which we had, you must have got a basic idea why does polymorphic mimicry evolve. The second question is, can variant exhibit non-morphological differences. All these butterflies, they mimic different species. They, because they are of the same species, but they have different phenotypes. So these are called morphovariants. So I will be using this term morphovariants again and again. So I hope there won't be any confusion. Third question is, can assortative mating exist between morphovariants? This is a very pertinent question because morphovariants, they are of the same species, so they should mate among themselves. But is it possible that they stop mating among themselves? So if they stop mating, so this is a transition phase which may lead to evolution of a new species. Can morphovariant be genetically distinct? We we hope they should not be genetically distinct, but we test this hypothesis in this study. And
Srishti, mute
please,
your mic. Please continue.
and how much genetic difference is required for considering an animal to be of different species? This is a very troublesome question. I have attended many different workshops and seminars, and I have asked this question to the experts coming from international institutions, from USA, from UK. But every time I get the answer which is unsatisfactory, I am still not satisfied with the answer. I am also not satisfied with the norms, with the principles which are there, because I think not a single taxonomy will be satisfied with its answer, because the answer is quite consistent and contentious also, because the percent is varies from taxa to taxa, and sometimes we also discover exceptions. So it is very contentious question. And the last question is, if there are non-morphological differences and assortative mating, can the morphovariant be considered different species? So all these questions, all these issues, we will be trying to look at individually one by one in upcoming discussion. So the site of study I chose, it is Hazaribagh Plateau region. So I chose, why did I choose Hazaribagh Plateau region? It is part of Chota Nagpur Plateau, which and it is interesting to know that this plateau is the ancient most landmass in the entire world. So we are Indian, but we don't know that Chotanagpur Plateau has ancient origin that dates back to Gondwana period. And Chotanagpur Plateau comprises of the oldest landmass exposed to air. And this study was published in PNAS. This is a famous journal for the students. Like Nature and Science, PNAS is also very reputed journal. And this study was published in 2021 in PNAs, that Chota Nagpur Plateau is the ancient most landmass. So the forest which is there in Chota Nagpur Plateau, it is the ancient most forest, forest not only in India, but in entire world. It is ancient. It is older than Himalayas. It is older than Nilgiri Hills. It is older than Northeast. Most of the scientists working on taxonomy, they are studying fauna of Northeast India, fauna of Himalaya, and fauna of Nilgiri Hills, the Western Ghats. But the Chotanagpur Plateau is ignored altogether. So I took this study for more exploration due to all these regions. So I selected few sampling sites at Chotanagpur Plateau, particularly Hazaribagh Plateau. So Hazaribagh Plateau, it includes different districts of Jharkhand in the northern region: Chatra, Bokaro, Giridih, Kodarma, Ramgarh, and Hazaribagh. So these districts come under Hazaribagh Plateau region, and I created this map using the gadget of Hazaribagh. The gadget, it dates back to 1916. It was prepared by Britishers. So I uh, means retrieved that gadget from library, and I created this map by the help of a friend. So this is the outline of Hazaribagh Plateau. So I selected these 10 densely forested sampling sites, and uh, I used basic methods, basic entomological tools for collecting insects, and I did further analysis like identification, characterization of taxa under different types and subtypes. And how do we characterize taxa? There are keys. If there are keys, we create our own key. If the species is new, if the taxa is altogether new. Then we did data analysis and some biodiversity quantification using Guinea-Simpson index and Shannon Weiner diversity index. So this is the initial part of this study. So we found like 174 species, and we found the diversity was moderate in nature. And out of these 174 species, we found Betzian mimic to the highest in number. When we resolved this Betzian mimic, we found different types of bits and mimics based on the type of model they are mimicking. There are ant mimics, there are wasp mimics, there are honeybee mimics, there are bumblebee mimics, there are velvet ant mimics, so and so forth and so on. So like there are variety of mimic. We categorized it based on the type of model it is mimicking. When we resolved it further, because the ant mimics were highest in number, so we resolved it and we found that there are spiders which are in largest number in mimicking ants. So we selected two different taxa, two different genera from this. Arachne is the group of a spider, particularly the modern spiders. They belong to the family Arachne. The bigger group is Arachnida. So we selected two ant mimicking a spider from this Arachne, and the first group is called Myrmeracne. So the genus Myrmeracne is the largest salsicid genera. It has 186 species globally, and 23 are found in India. And out of 23, only 6 are well characterized. And they exhibit Wetson mimicry, and they resemble ants. So the species which I discussed in the beginning, the story, this species was actually Myrmeracno melanocephala, and the ant which it is mimicking, it is called Tetraponera nigra. This sketch, this painting has been drawn by me. Yeah, as you can see, it is also in my background while I am talking. You can see through my camera. So it mimics Tetraponera riponigra. Why did I study Myrmeracne when it is already known? So I will comment on this later. So I performed a standard taxonomic protocol to study this species. So I dissected it. Particularly, this structure is important. This is the structure of female genitalia. So why did I dissect female genitalia, and why did I study Myrmeracne melanocephala or Myrmeracne? I will come on it later. So this is the exuvi, like snake, uh, uh, like they undergo ecdysis. They exhibit molting. They shed their skin. We call it in Hindi. We call it केचुली. Like सांप है जो केचुली बदलता है. Likewise, insects and many spiders. Spiders are not insects. Those people who are new, they should know that insects or spiders are different. Insects have 6 legs, and spiders have 8 legs. So, like snakes, spiders also change skin. So this process is called molting. So this is and the cachuli is called exuvi in English. And we also studied male. So we are more interested in female. So why we are more interested in female, I will come on that. And this is the model. So we were conducting this study just to facilitate, to provide information for upcoming study, which I'm going to discuss. So this was the background study, but while conducting background sampling also, we came across many interesting behavioral patterns and many interesting discoveries. For example, this is the D-elite queen. We discovered D-elite queen of this species for the first time, and we described it. Similarly, we also found this. This is a live insect. It is carrying dead. This is the live ant. It is carrying dead ant. This is Tetraponera only over its back, and this gait, this posture is mimicked by the male spider, and this is the normal worker Tetraponera, and the female spider, it mimics this form. So you can see how behavioral mimicry is also there. So this particular study, some behavioral, we also conducted some biogeographical mapping, and we found that this species, that Myrmeracno melanocephala, it is only limited to few states of India. But after this work was published, I found that this species is found everywhere in India. So the study is lacking, and the scientists, they write in paper that mimics are rare in nature. No, I don't feel mimics are rare in nature, because this particular species, I went to Ajamgarh, I found this species in Ajamgarh also. I went to Varanasi, I found this species in Varanasi also. I found this species in Ranchi, I found this species in Hazaribagh. This species is also found in our college campus. So how we can say that this species in rare in nature and mimics are rare? No, these are not rare. The studies are lacking. That's why, due to paucity of study, people say that this is rare. All these issues have been discussed in this chapter, this paper, which was published in 2021. So this was the background study only. The main study, it focuses on the genus Myrma plata. So Myrma plata is a small genus. It comprises of only 5 described species, and it was created from Myrmeracne also. The previous genera we studied, Myrmeracne, it was the it was the older genera. From that genera, Myrma plata was carved out. Why? Because the female genitalia, I told you, I discussed that the structure of female genitalia is important. So I am now coming on it. The structure of female genitalia in Myrma plata is different from the structure of female genitalia in Myrmeracne. So a new group was created, Myrma plata, and it has only 5 described species. And Myrma plata platyloidot, it is red in color and mimics ant called Echophilus maragdina. It is the common red ant. You must have seen a red ants running over a mango tree. So this is the same ant, Echophilus maragdina. And like I discussed about polymorphic mimicry, I have found that Myrma plata platyloidots is also reported to exhibit polymorphism. A study was performed by Rini Borges ma'am from IISc Bangalore in 2007, where she studied different morphs of Myrma plata platyloidots. Her study focused on the mate selection, so it was not taxonomic study. So I have also conducted taxonomic studies. So different color mobs of Myrma plata have been reported to mimic different models in habitat besides Enothera, Echophylla, Smaragdina. So this is termed as polymorphic mimicry, which I have already discussed in case of papilio butterflies. So this spider I found at Hazaribagh Wildlife Sanctuary. There is a tiger trap. Tiger trap is a structure which is in many old forest. It was the tiger trap in Hazaribagh Wildlife Sanctuary. It was built by local king many years back, like 500 years back. We don't know the exact timing, but people say this is very old, around 5 year old. So near that tiger trap, there is a huge tree of Nyctanthus arbotristis. We call it. We also call locally as harsingar. Bengali call it shuli flower. Shuli flower. It has got mentioned in different poems of Rabindranath Tagore. In South India, in Maharashtra, this flower is called Prajakta, and some people also call it Parijat flower. And it is very common in North India, and particularly it has relevance during Dashara. It is used for worshipping Durga Ma, and it indicates the arrival of Aswin month. You must have seen its flower. So on this tree also, what I found that beneath this tree, there was like a huge dominant colonies of a black ant. It is called Camponotus compressus. It is the same ant which visits our home. It is the bigger ant. It also bites. So this is the common black ant. So while screening the tree over its branches, I found entire population of black colored spider. So I thought this may be some new species. But after I studied thoroughly, this is my material method and sampling. I sampled for a whole year, because if you plan to study behavior, you need live specimens. So for live specimens, you have to do a large scale sampling. And large scale sampling cannot be performed in one go, because what will happen that it will lead to extinction of that population. So I visited the forest um, like 1, 2, or 3 visit every month, and I collected few spiders. I did not collect whole because I do not want that population to become extinct. So after collecting, I performed some taxonomic studies in the beginning. Then I performed behavioral studies like egg laying, molting behavior, feeding behavior, photokinetic responses, response to starvation, response to hypoxic condition, coercive behavior, coexistence with model. So because this is a big study, so I will be discussing only the important part, because the time is not sufficient. Otherwise, it will take 2 hours to discuss everything. So this study is like a monograph. So in the beginning, we performed morphotaxonomies, and we found that this is the typical reproductive structure of female in Myrma plata. So, like I discussed in the Myrmarachne, here you can see the female genitalia has loop-like structure. This is called spermatheca in Myrmarachne, but in Myrma plata, the spermatheca is bulb-like. You can see bulb-like structure. We also studied its ultrastructural detail, and we found that there are internal projection in the spermatheca. This is the male, this is female. The male has this prolonged chelicerae, it is called chelicerae. And the male, this is the reproductive structure of male. Those who are new to spider, it is very interesting to discuss a fact with you all, that like different insect, they do mating for reproduction. They mate among themselves. The mating of spiders involve carrying the spermatophore over their labial pulp. This structure is called labial pulp, and it is present nearby their mouth. They keep they store the sperm in this structure, and they transfer sperm using their labial pulp to the genital pore of the female. So this is very common among many spider, particularly salty seeds. Salty seeds are called jumping spiders. And the spider which I am discussing with you all, this is also jumping spiders. So this is how they reproduce. So this is the reproductive structure, that is structure labial pulp of the male, and this is the structure of female genitalia of the. This is the genitalia of the female spider, Myrma plata. So it is consistent with the red form. This is the black form you can see, and I already discussed that red form is well characterized, but black form is not well characterized. It is very common practice in taxonomy that people are more interested in identifying new species. If they find that this particular sample they have collected, the particular material they have found somewhere, it is the morphovariant of already known species, they are disinterested and they do not study it further. But I took a risk to study a morphovariant thoroughly. So this study is red typical form versus melanic morphovariant. So I already discussed the red spider. This spider has been sent by my friend Dr. Stuti from Pondicherry, and it is very common in South India. And I could not find the red form in Chotanagpur Plateau as well as in Bihar, where I am working right now. And it mimics the red ant Echophilus maragdina, and the black spider, they mimic another ant, that is Camponotus compressus, as I already discussed. So what do we propose? We propose that a darker coloration would have been naturally selected in this isolated population of Myrmaplata platyleoides as an adaptation to coexist with Camponotus compressus, which are also black in color and are abundant around the spiders. We studied different aspects of behavioral patterns, and we studied food preference, courtship, mating choice, and all these are similar to that of the red form. So what are the differences? The differences is the number of eggs laid by them. These lay more than 20 eggs at a time, but I have read in papers and literature that the red form lay around 12 to 15 eggs only. This is the distribution of this particular spider in India, and you can see that the red form is distributed across different locations. And we have found the black form particularly in here in Jharkhand. And although in literature we found a red form is also found in Bihar, but I could not find a single red form. And from Jharkhand, I have discussed this species for the first time, that too a different morphovariant, that is black. Then we performed some observational studies on molting behavior. And what I found that the red form, the typical form, what again and again I am telling, speaking about typical form. So what is a typical form? So typical form is that form of a species on which the description of that particular species is based. Hope it is clear. So the typical red form, when it exhibits molting, what happens? That their size increases, but they do not increase in their breadth. Like they are altogether slender, because the ant is also slender. But what you can see in this black ant, that there are different subcasts. There is the smaller minor form, there is major form, and this is the medium form, this is the major form. So you must have seen these ants. Some are smaller in size, some are bigger in size, and all these are of the same species, but there are different subcasts. So interestingly, what I found in this species, this morpho variant, I am saying species again and again, but this is a morpho variant of the red form only. So what I found that after sixth molt, what happens? Its breadth also starts increasing significantly. So why such bulking is observed? So my hypothesis was like, it may be possible that the seventh form, the seventh molt, it is mimicking the minor form. The eighth, this is the sixth, this is the seventh. The seventh malt, it is mimicking the medium, and the eighth malt, it is mimicking the major ant, the major subcaste of this ant, Camponotus compressus. So but it could be a chance observation, it is also possible. So what I did, I reared all these malts with different subcaste, and I found that the smaller one, it tries to attack only the spiders which are smaller than its size. So by increasing the size throughout molting, what they are doing, they are getting survival benefit from for themselves against the attack of aggressive ants. So the larger molt, they are not attacked by the smaller subcaste of this ant. And we have named this mimicry as temporal myrmecomorphy. Temporal is related to time. And temporal myrmecomorphy has been proposed to be a protective mechanism used by harmless mimic against the attacks of aggressive models, so that it can coexist with the model. Yeah, one thing should be noted, that although these spiders mimic these ants, they always maintain a safe distance from these ants. From far by, these ants will think, yeah, these are our own conspecifics, they are member of our own population, they are also ants. But when they come close to the ant, because ant also have a strong olfactory sense, and during daytime also they have a good vision, so they can recognize these spiders as imposters. Imposters means Behrupia. So they will think these are Behrupia, these are imposters, and they will attack on them, they will kill them. So they always maintain safe distance from them. And by increasing their size during molting, they are getting survival benefits against the smaller subcaste of their model, and that is Camponotus compressus. We also observed exuviation in male. We, like Kechuli change Karna, I told that snakes also change their exuvie. So we observed this live, how this particular male, this is a male, it is changing its Kechuli, its exuvie, and how the new skin is growing. Here you can see the chelicerae, it is white in color, and within 15 minutes it becomes completely black. So we saw exuviation, means in situ, on time. So sorry, it is a ex situ. We also have many in situ observation. This was ex situ. When we say in situ, it is like on site. When we say ex situ, it is away from the site. So these are ex situ observations. So this work was published in a journal, Animal Biology. This is one of the oldest journal of zoology in this world. And it was, and one of the photograph from our paper was also featured on the cover page of this particular journal. And when this work was published, it was studied, and I also got critical appraisal by various people. And one of the papers cited this work, and they questioned my finding, and they suggested that the morphovariant which I am studying, it could be a new species altogether, because it is exhibiting so many dissimilarities with respect to the red form. So we decided to conduct molecular studies. So there are some issues with Mirma plata. This also prompted me to conduct molecular studies. So what are those issues? There are reports of morphovariant in Mirma plata platyleoides, as I already discussed.
और कितने स्लाइड्स हैं?
सर कुछ और हैं स्लाइड्स.
कितने मिनट और लग जाएंगे?
हाँ सर, अगर जल्दी करना है तो आई विल कंप्लीट इट इन 5 to
कर
10
दीजिए.
minutes.
1 घंटा जनरली सोचते हैं और करिए, मतलब 10-15 मिनट में हो जाएगा ना? क्या
जी
ऑर्डर?
सर,
जी
हो
जी
जाएगा
जी
सर.
जी 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 कर दीजिए, है ना?
Okay. So so we decided to study molecular taxonomy also. So what I did, we isolated DNA from the black form. I compared. Then this is the raw images, the gel image of the PCR product. So we used these primers. Here you can see. Let me turn on the pointer. Yeah. So these are universal primers. We use it in molecular taxonomy mostly because there are students. I would like to tell them that we used mitochondrial cytochrome oxidase 1 gene sub. This is the mitochondrial DNA. This gene is present in mitochondrial genome, and we amplify this particular gene and we sequence this gene and we compare sequences from different species to construct phylogeny and to perform to construct molecular barcodes. So this is standard, means this is standard among insects, and this has also been used among spiders. And as a backup, we have also, as an extra evidence, we have also sequenced the one of the nuclear gene also. This is called 28SD9D10. 28, this is part of 28S rRNA gene only. This is small part. So we have also sequenced it, but we could not do any analysis because the D9 D10 sequence of other species is not available online on NCBI. So we stick to CO1 only. We submitted these sequences at NCBI GenBank, and this is how the DNA barcode look like. We construct DNA barcode. We actually deposit the sequence on BOLD database. It is the repository of the DNA barcode of all the species worldwide, and this is how the barcode will look like on its website. So if from India, we have contributed the significant amount of barcode for this species. One unspecified submission was also there. One submission was from Singapore for the red form. So we took the specimen submitted by Singapore, its barcode, for comparing with the black form studied by us. So these many studies of these many species of Myracne uh, genus and Myrma plata were taken into consideration, and we first of all performed multiple sequence alignment, and we found that the black form is genetically similar to red form, but there are some differences you can see, which I have indicated by red arrows. Based on these sequences, I have constructed a phylogenetic tree. Why I have used Myrmeracne also, why I have used Myrma plata, hope it is clear now. Because Myrmeracne is the parent genera from which Myrma plata was created. So we have performed a phylogenetic study, and when we constructed phylogenetic tree, we were surprised to find something that it is fine that these are Myrma plata, this is the black form, and this is the red form. So they are coming together, so that's fine. But this is also Myrma plata. This is another species, Van Lessie, and this is also Myrma plata platyleoides. They should be together, but they are falling far apart. So it raises question on the authenticity of this species, Myrma plata Van Lessie. So, and again, you can see it is showing a similarity with the DNA sequences of Myrmeracne lembirensis, which is other species of Myrmeracne. So Myrma plata vanellesi is genetically more closely related to Myrmeracne lembirensis than Myrma plata platyleoides, and we will find how. So first of all, I estimated evolutionary divergence to compare the red form and the black form, and we found a significant divergence that is 3.84%. So And this is the molecular divergence we found. And as I already discussed, that Rini Borges ma'am from IIC Bangalore, she found associative mating also among different morphs of Myrmaplata platyleoides. So what happens? There is a trait, it is called magic trait, which is related to evolution. So what happens that there are some traits which lead to difference in their genetic sequences, which in turn can be used to quantitate evolutionary divergence. And this also lead to the assortative mating, that is different morph will not mate with other morphs. Like black form will not mate with red form, black will always mate with black form, red form will always mate with red form, although they belong to the same species. So such trait ultimately lead to speciation, and this is the transition phase. This is called magic trait, which ultimately lead to evolution of a new species. So based on our observation and previous work of Rini Borges ma'am, we conclude that polymorphic mimicry in this species, Myrmaplata platyleoides, is a magic trait, because we found evolutionary divergence of 3.84%, and she also found existence of associative mating. So these two criteria is being fulfilled. So this trait is a polymorphic trait. And also, why don't we consider it a new species? Because 3.84% is not considered significant among spiders. Recently, a work was published in Nature Communication, in which they have shown up to more than 4% evolutionary divergence between the morph of the same species. So we are not considering it a new species altogether. Also, the reviewers, when I submitted a paper, some of the reviewer, they have commented that cytochrome oxidase 1 gene, based on single gene, the phylogenetic tree, that it, if we construct a phylogenetic tree based on a single gene, it is not robust. We should consider a large number of genes. We should concatenate it and create a robust phylogenetic tree. So I am doing that right now. So, and coming back to the Myrmaplata vanellesi, it was, as you can see, it is clustering with other species rather than it should cluster with Myrmaplata platyleoides. What I found in, I performed meta-analysis and during literature survey, what I found that this is the Myrmaplata vanellesi. Its male reproductive structure, it is identical to the male reproductive structure of the Myrmarachne lembirensis. And it is very surprising to note that this species was described on male only. The female is missing, but in this is the female is known. The end of female genitalia, it looks like the genitalia which I discussed in the beginning of this presentation, that is Myrmarachne. So it shows that this species is artificial and it should be brought back to Myrmarachne lembirensis. Even if these two species are not same species, it is not certainly a Myrmaplata, it is certainly a Myrmarachne, because its male genitalia is identical to the male genitalia of Myrmarachne lembirensis. So this is the slide just for comparison. Myrmaplata genitalia, its spermatheca is bulb shaped, and Myrmarachne, its genitalia is like, this is loop like. Here you can also see it is loop like. So it is not Myrmaplata, it is Myrmarachne. So this work was also published recently in a small journal, and this is the summary of all the interesting discoveries I discussed with you. This is the graphical summary, and this is the summary in text. I will not go one by one, just I will point out on the terms written in red, the key words which we include in papers. We call it keywords. Keywords help in searching, discovering a particular topic on internet. So I will just read the keyword. So these are the major finding of this work: temporal myrmecomorphy, significant evolutionary divergence among morphs, magic trait, conspecificity between two different species which were considered to be the same species, and the morphovariant could be the important model. And these are the final conclusion: mimics are widespread in nature and can be fantastic subjects. Morphovariants are important model system in ethology and evolution, and they should not be ignored. There is red queen effect when we talk about temporal myrmecomorphy. So what is red queen effect? Queen effect is a term used for arm race. What is arm race? Like right now, very recently, I don't think it has ended till now. Also, the conflict is going between India and Pakistan. Pakistan has killed many of our citizens, innocent people, innocent tourists. They were traveling to Kashmir, and in retaliation, the India has attacked Pakistan. And when India attacked, Pakistan is also attacking. So this is like, I will attack, you will also, they will also attack. Again, we will attack. So it is a like a chain event. So such event is called Red Queen effect or arm race. So it is arm race in real, like we are also having missiles, they are also having missiles, and we are attacking one another due to some conflict. So this is called arm race. And here also arm race is going on. If the model is evolving, mimic is also evolving, and mimic is trying to adapt to the model so that they will not be attacked by the model. So this is a kind of red queen effect. And polymorphic mimicry is a magic trait, as I already discussed. And explicit delimitation rules are inconsistent and contentious. Why inconsistent? Because I discussed you like there are different morphs which have more than 4% evolutionary divergence, as published recently in Science Communication, but they belong to the same species. But in many insects, what happened, like there is a law called de Barrow's law. So de Barrow's law, it tells that if there is more than 3.5% difference between the genetic material, the material should be considered a different species altogether. And this applies to many insects like Hemiptera sap-sucking insects. So it applies to them, but it does not apply to spiders, which we are studying. And the studying behavior in taxonomy is important because we could find the differences among morph, the behavioral differences only when we studied behavior. We found associative mating. It may be possible it will become a new species in future, or it is indeed a new species and we have not performed enough analysis. So I am performing more analysis. Let's see what happens. Again, it proves it is morphovariant or it is the different species. So more interesting discoveries are yet to come, and taxonomic discrepancies are there, and it needs an integrative approach. Like this is I have taken from a recent from a paper where they have shown that the future of taxonomy is integrative. It is based on the evidences from multiple disciplines like molecular biology, geography, ecology, reproduction, morphology. But again, you can see here this diagram I have taken from this paper. They have ignored behavior. So behavioral studies are also important and they should be taken into account. So what's next? So I am right now studying the mechanism of reproductive isolation among different morphs of Myrma plata platyloides. I am also trying to create phylogeny of Myrmarachnum, Myrma plata using sequences from more genes. I want to find the molecular basis of polymorphism among Myrma plata platyloides populations and many new models, because I have described I have found 175 mimic model pair from Hazarebaig plateau region. So there are many new model which can be used to study mimicry. So these are some recommendations from my part for the people who consider taxonomy as an old science. People are running behind um, molecular biology and immunology. All these things are important because these are related to human health and human welfare. But we should not remove taxonomy from entire picture because it is at the base of all these discipline. Because if you stop discovering new species and describing new species, correctly describing species, what will happen? We will go out of our the information of our own bioresources. And also and science should not be anthropocentric, because this is our present view that everything which we do in science, it should benefit human beings. No, there are other animals also, they also have life, they also feel pain. So the science should not be completely anthropocentric, and this is also one of my recommendation. And the last recommendation I would like to tell, because I will not go through each of these bullets because we don't have much time, but I would like to stress upon this particular point, that is mainstreaming taxonomy. So teaching taxonomy without emphasizing that it is old, classical, exclusive or outdated science is very important, because it is not true. Because taxonomy is a modern subject, it is not a classical old subject. So students should be taught that it is also a modern field of science like biochemistry, molecular biology, immunology. NCRT textbook, they present misleading image of this field. And how do I know? I have worked at NCRT. I am working at a science department also. I was involved in curriculum development, and I found that the NCRT textbook, they create misleading image of taxonomy because the experts they invite in their workshops for designing curriculum, they are from the same field. They mostly invite people from DU, colleges of DU, and some people from JNU, and they are only versed in molecular biology, and they don't have any know-how of modern taxonomy. That's why the content on taxonomy is lacking in our textbook. Also, for studying taxonomy, particularly zoology, they rely on Kotpal, and you should know that the Kotpal book has not been revised for last 30 years. In the name of revision, they have just changed color of the text, added some color and colorful imagery. That's all. The main content has not been revised for last 30 years. So redesigning taxonomic pedagogy recommended to present a clear picture of taxonomy as an integrative discipline and encourage students to pursue higher research in taxonomy. So moving away from convention, I have also used nanotechnology in taxonomy, and this paper was published in 2022. And I have got some critical appraisal from my work because I am a trained molecular biologist, but I have switched to ecology and evolution very recently. So I'm still learning ecology and evolution, and I am finding this discipline fantastic. And because everyone is doing molecular biology, so there should be some people who are doing evolutionary biology, ecology, and taxonomy. So this is Rini Borges, ma'am. I cited her again and again during my lecture. She appreciated my work. I also presented my work to her in one of the conferences. I also got appraisal from a famous scientist from National Institute of Genetics, Mishima, Japan. He's a famous evolutionary biologist, Jun Ketano, and he suggested me that the model which I'm using, it can be a great model for studying speciation. And I have also got recently this work was featured in BioTales. This is a initiative by uh, TIFR, and by scanning this QR code, you can read this story. This is the story how I got interested in this spider and what and where this story is leading me to for more discoveries. And I got some critical comments from Rini Borges ma'am again for these articles. And some of my work has also keep on featuring in local and national newspapers. And my work also came on national television on Now Bharat Times. And I'm also associate editor of a magazine, Indian Entomologist, and it is free of cost. And any student, anyone, even he is not an entomologist, he is an insect enthusiast, they can submit their article, their even there are photographic contests right now going on in Indian Entomologist. The deadline is not over. They can submit their photographs, the insect photographs, and it will be published in the magazine. And interestingly, the magazine is published by the Entomological Society of India. It is the oldest scientific society in India. And this is my lab. This is the sketch of my lab, and this is botanical garden where I conduct in situ behavioral studies. This is my lab where I conduct biochemical analysis and in situ studies. And this is, you can see the Son River it flows nearby our campus. So our campus is a kind of biome where different insects they keep on coming because surrounding areas are farmland. So our campus, it provides a natural habitat for all these insects and spiders, though they frequently visit our campus, and it provides me enormous opportunities to look into their life, natural history, evolution, and taxonomy. These are the key areas I am working on right now: nanotaxonomy, chemical ecology, biodiversity, behavior and survival strategies, applied entomology, and evolution. And all the images you can see here, they have not taken from internet, they are from one or other paper or some unpublished data of mine. So I would like to acknowledge few people because I'm working in a far rural area in Bihar. So without collaboration, it is not able to pursue research at this level. So I have collaboration with my previous supervisor, Subhash Yadav sir from AIMS New Delhi, Shashank sir from IRI. And I also take help from some renowned arachnologists like Kaler sir. And my friends also help me. And also, this is the stuti whose name I took again and again during my presentation. She's a great entomologist and she frequently helped me. And recently I got opportunity to get training on experimental evolution on Trilobin castanium at the lab of Deepa Gashi ma'am at NCBS. So I'm also trying to establish these beetle lines in my lab, and I'm working on experimental evolution using these Trilobin castanium. This is floor beetle, normal floor beetle you have seen in floor. आटा में जो छोटा छोटा कीड़ा होता है, this is the same insect I am working on right now, and I have got the training from NCBS to work on this. So thank you, and if you have any question you can ask, because I would rather have questions that can't be answered.
थैंक यू बहुत अच्छा से आपने इंटर्मोलॉजी में इंटरेस्ट जगा दिया है। ये सही बोला कि बहुत इग्नोर्ड सब्जेक्ट है बट इम्पोर्टेंट है ही।
और सर, one more thing I would like to say, actually I was doing MPhil PhD from JNU, but I quit PhD in between, and after working in IRI I completed my PhD after joining here as faculty.
ओके ओके
Yes.
ओके
So because I was interested in molecular biology, but I did not like repeating same experiment again and again and again. Everyone was doing same thing in entire school of life sciences, and I had some personal reason also. And I was always fascinated with forest. And I read interview of Dr. Raghavendra Gadakar at IISc.
हाँ गदाकर हाँ
Thakur sir, and I was very much influenced. He was a molecular biologist, but he left molecular biology and started studying the wasp behavior. So after reading his interview, it instilled some, like it was like igniting some interest. Means that interest was hidden inside me, because when I took zoology as a subject, I liked studying taxonomy, I liked studying evolutionary biology. But because in MSc we did not have ecology and evolution in our university, so I studied molecular biology. So I took molecular biology for PhD, but later I dropped it and I started working in this area. And now I find that this area is full of opportunities. And recently I have written an intramural grant also, and I hope that I will get that.
थैंक यू नाउ प्लेटफॉर्म इज ओपन फॉर क्वेश्चन